Hello, my name is Jancarlos Gomez, and today I'll be presenting the use of electromagnetic fields in stem cell engineering. Electromagnetic fields are a form of radiation that can be attributed to the acceleration of electric charges. These fields can affect cells when cells are exposed to them. A variety of factors such as electromagnetic fields, radiation, biological conditions, stress, trauma, injury, chemical reactions, and more can affect the functionality and characteristics of stem cells as well as change their phenotype. These factors can either positively influence a stem cell by increasing regeneration rates, ability to differentiate into desired cells, obtaining desired growth, and homeostasis, or these factors can negatively impact the stem cell's development, make them carcinogenic, degenerative, have an unwanted differentiation, or cause pathological growth. Thus, when utilizing electromagnetic fields, it is crucial to utilize extremely low frequency electromagnetic fields to yield desirable cell outcomes because if a frequency that is too high is utilized, too much reactive oxygen species can be generated, and this can damage various cellular structures. Reactive oxygen species are a type of molecule that contain oxygen and easily react with other molecules in a cell, causing them to become injured in their proteins, lipids, and nucleic acids, eventually leading to the cell's death. Electromagnetic fields can influence stem cells in a variety of ways. Utilizing an electromagnetic field on a somatic cell, we can turn it into an induced pluripotent stem cell, as shown in the image on the right. Depending on the frequency, intensity, and duration of the EMF exposure to the cells, it can cause an increase or decrease in the proliferation rate and in the viability of the cells. EMFs can also alter the cell cycle and make cells die sooner or later by modifying the cell's survival signal. This can be used to delay apoptosis, which is a form of programmed cell death. EMFs can also alter the morphology of cells, which is the shape, size, structure, color, texture, and pattern of cells. EMFs, as previously mentioned, can also cause reactive oxygen species to be generated, which can pose as a danger to the stem cells. Several studies have indicated that stem cells influenced from EMFs can increase their marker expression and probability of differentiation into the following cell types: cardiogenic, neurogenic, osteogenic, and chondrogenic cells. However, a few other studies have shown the opposite to be the case. Depending on the day of exposure, condition of the culture, frequency, intensity, and duration of the EMF exposure, osteogenic and chondrogenic differentiation and markers could decrease as well. Thus, more studies need to be conducted. Electromagnetic fields have the ability to increase the amount of calcium ions present in the microenvironment of mesenchymal stem cells. These calcium ions are capable of activating the focal adhesion kinase and Rho GT-pases signaling pathways. Through this activation of the signaling pathways, mesenchymal stem cells can have their healing performance increase by improving their migration capabilities. When stem cells are implanted into a targeted area, some cells may have a low migration capability, making them a poor candidate for the healing process. Since these cells may end up in other parts of the body, stem cells that are not properly migrated can cause issues such as chronic inflammation to occur while also not contributing to the desired healing process. We can observe in the figure on the right that between 7.5 Hz and 50 Hz at 1 microtesla, the number of migrated cells increased compared to the control group that had no EMF exposure. At 75 Hz and beyond, diminishing returns occur, and we can observe that the amount of the frequency for the EMF becomes too great and the benefits obtained start to decrease. Since we notice that at 75 Hz we obtain less cells migrated than at 50 Hz, this can also be due to the amount of magnetic flux density present. If we use a smaller magnetic flux density, we can utilize a higher frequency. Pulsed electromagnetic fields are when EMFs are created in non-continuous bursts. Utilizing this method of electromagnetic radiation, brief periods of strong magnetic pulses can be utilized on abnormal mesenchymal stem cells to make them return to homeostasis. Previously, a team was able to treat rheumatoid arthritis by targeting aberrant mesenchymal stem cells. The effects of pulsed electromagnetic radiation influence the charge on the membrane surface as well as the membrane's potential. Pulsed EMFs can alter cell membranes, nucleic acids, and even the bioelectrical phenomena generated during the cell-to-cell communication process. Thus, utilizing this technology is a major advancement in getting stem cells to act in a way we want them to. By altering the intracellular calcium concentration and restoring the levels of potassium channels and mesenchymal stem cells, we can restore natural balance between calcium ion flux and sodium potassium ions. In the image on the left, we can observe that prior to pulsed EMF treatment, the performance of the immune system was hindered due to abnormal mesenchymal stem cells. But once pulsed EMFs were utilized on these mesenchymal stem cells, proper immune system function was restored. All in all, the science of using electromagnetic fields for stem cell therapy is still relatively new, and a lot of research needs to be accomplished. The use of extremely low frequency electromagnetic fields and pulse electromagnetic fields has shown to be viable and beneficial for stem cell therapy. However, there has been a few studies that have shown contradicting evidence, but this can be attributed to studies not being uniform when selecting cell samples. Characteristics such as age, cell density, and cell type are neglected. Other factors such as the testing environment and the intensity and duration of the EMF exposure is not even throughout the studies. Future studies should be conducted uniformly and repeated over several different samples to obtain concrete results. Most studies appear to choose their EMF exposure, cell samples, and testing environments at random without giving much explanation as to why. More information on the current state-of-the-art of electromagnetic fields and stem cell therapy can be viewed in the links on the references page. These past studies and results are very intriguing. Furthermore, as we can observe on the image on the right, EMFs cause an increase on the calcium ion, which has shown to provide many benefits in stem cell therapy, as previously mentioned in prior examples. Another note I would like to make is that studies should be conducted on other types of stem cells, since most studies have been conducted on only mesenchymal stem cells. Other stem cells may benefit from electromagnetic field exposure as well. These are the links to the references utilized for the creation of this presentation. Please make sure to check them out, since much information and studies on current electromagnetic field effects on stem cells can be found there. Thank you for watching this presentation, and I hope that you enjoyed.